We know that an elephant only gives birth to a baby elephant, and a mango seed only grows into a mango tree. But if offspring inherit their traits from their parents, why do siblings often look so entirely different from one another? Humans actually figured out how to manipulate this biological quirk thousands of years ago. Between 8,000 and 1,000 BC, early agriculturalists noticed variations in wild populations and started selectively breeding plants and animals to isolate specific traits. By repeatedly selecting wild cows that possessed desirable characters, for example, our ancestors successfully domesticated distinct lines, like the Sahiwal cow in Punjab. Yet, despite successfully breeding these animals, early humans operated on a surprisingly inaccurate assumption about how heredity actually worked. For centuries, the leading idea was the blending theory, the logical assumption that traits from a mother and father simply mix together in their offspring, exactly like blending two different colors of paint. There is a massive, inescapable math problem with this theory. If traits constantly mixed over time, any distinct characteristics in a population would eventually wash out, averaging every organism into identical clones. Curious, if heredity is just a continuous blending process, how does sharp, distinct biological variations survive generation after generation without turning into a uniform gray? It turns out, for thousands of years, humanity managed to successfully manipulate the architecture of life while holding a set of blueprints that were completely upside down. That contradiction persisted until the mid-19th century, when a monk named Gregor Mendel decided to test heredity using an entirely different discipline. Instead of making casual observations, Mendel brought statistical analysis and mathematical logic to a biological problem. To get reliable numbers, he needed a massive, perfectly controlled sample size. He spent 7 years cultivating garden peas in his abbey, meticulously isolating variables before ever attempting a single formal experiment. He focused on 14 true breeding pea lines. A true breeding line is a plant that has undergone continuous self-pollination until it shows completely stable, identical traits for several generations. Mendel took those stable lines and organized them into pairs that were similar except for one character with contrasting traits. He crossed plants with violet flowers against those with white flowers, or tall stems against dwarf stems. By building perfectly consistent true breeding inputs, he took biology out of the realm of observational guesswork and turned it into a rigorous mathematical testing ground. This diagram illustrates his first experiment, crossing a true breeding tall pea plant with a true breeding dwarf plant. According to blending theory, combining these parents should produce a medium offspring. But when grown, the F1 generation, every plant was completely tall, matching only one parent. The dwarf trait didn't blend. In this generation, it appeared to have been erased. To find out where it went, Mendel took those uniformly tall F1 plants and allowed them to self-pollinate, creating a second filial, or F2 generation. This updated lineage tree tracks that next step, revealing a biological shock. When the tall F1 plants reproduced, the dwarf trait suddenly reappeared in the F2 offshoots, perfectly intact. And crucially, absolutely zero plants in this second generation were of an in-between height. They were entirely tall or entirely dwarf. The fact that a physical trait could skip an entire generation, hiding completely out of sight without being altered or diluted, proved that the biological blending theory was mathematically impossible. By tracking thousands of these F2 plants, Mendel discovered that the reappearance of the dwarf trait wasn't a random anomaly, it adhered to a strict mathematical rule. This data visualization shows exactly how those thousands of data points sorted themselves. Every single time, the final count was exactly 3/4 tall plants to 1/4 dwarf plants, a precise 3-to-1 ratio. That 3-to-1 ratio confirmed that the dwarf trait was never destroyed or mixed in the first generation. It was merely a recessive trait, temporarily hidden behind the opposing dominant tall trait. This established a completely new set of rules. Heredity is not a soup of blending liquids. Instead, it is driven by discrete, unchanging units of information that are passed whole from parent to offspring. By demanding rigorous mathematical logic and massive sample sizes from ordinary garden peas, Mendel uncovered the specific rules of heredity, founding the entire modern branch of genetics.